All right, thank you, Sushmita, for the introduction. Thank you for everyone who organized for inviting me here. So I'm really excited to talk to you today about a project that I worked on during my postdoc with Alcus Price. And then I can briefly also talk about my future directions for my group if that's of interest. I'm obviously recruiting graduate students and postdocs since I just started. So today I'm going to talk about inferring causal cell types driving human disease and complex. So first, I want to convince you guys that inferring causal cell types in tissues in disease is a very important goal. And that is because from GWAS alone, it is very difficult to infer anything beyond the level of the variant being associated with the disease. So for example, it does not tell us anything about the cell type level mechanism that may be at play, or the genes even that mediate this genetic susceptibility. And so ultimately, once we are able to infer the causal disease tissue or cell type, it can help us in one, describing the true biological mechanism underlying disease. Two, it can help us to select the cellular context in which to perform follow-up experiments. And three, it's important for determining the physiological target of pharmaceuticals if they were to be developed in order to treat the disease. So there's been many years of genome-wide association studies that have identified thousands and thousands of variants that are associated with complex traits and polygenic diseases. And there have been many methods that allow us to sort of co-localize either these genome-wide significant variants or variants in general at any statistical threshold with different types of functional genomics data. So here I'm showing, for example, you can look at gene expression, you could look at chromatin accessibility, at transcription factor binding, and these flavors of genomic data, as you know, come in different flavors of tissue, cell types, and single cell resolution data sets. And in fact, for most complex traits and diseases, associated cell types or tissues have been identified. And for this talk, I'm going to use cell types and tissues interchangeably. So for example, using chromatin accessibility, the authors that developed stratified LD score regression found that bone chondrocytes are the most strongly associated cell type for height. On the other hand, if we were to use gene expression data, specifically EQTL data, so meaning we have gene expression, not only gene expression, but paired genotype data across those individuals, this method called RTC colloq was able to identify that the frontal cortex of the brain was the most strongly associated tissue for schizophrenia based on the tissue and cell type level data that they had, which honestly was mostly from GTEx. Now, citing previous work from myself during my PhD, using in silico predicted tissue-specific or cell type-specific enhancers, we were able to find that CD4 Th1 cells are the most strongly associated cell type for asthma. And with this plot, I kind of want to remind you guys of what like Manhattan plots look like for GWAS. And so, of course, we see peaks of these associations, which are composed of correlated cell types. And so, sure, maybe we can identify the most strongly associated cell type, but at the end of the day, we don't know if the cell type is causal because it can just either tag better, for whatever reason, better data quality, the true causal tissue, and therefore lead to a more significant result. And so this warrants more careful methods development. So again, to reiterate this point, there's a high degree of general correlation of many different genomic features across cell types. So for one, if we look on the left of this figure, we know that gene expression is highly correlated across cell types and tissues, for example, in the GTEx data set. And so you see like that one big module for brain tissues, but even if you look at the other tissues, which represent very different parts of the body, there's also a very high degree of correlation. This correlation also exists at the level of co-regulation. So now we're talking about genetic effects on gene expression, the EQTL effect sizes, essentially. And so there's a very high degree of co-regulation, which means any method that's using EQTL data to infer associated cell types is going to be confounded, or at least it will be a little bit complicated to infer what might be the causal cell type from that data without carefully modeling what is the correlation structure in this or covariation structure across these cell types. So with an example, I want to show how co-localization of eQTLs with GWAS variants can help us identify disease-critical genes and tissues, but also why it gets kind of complicated. So let's imagine in red here, this is a disease-associated variant from GWAS. Now let's say in brain tissue, we see that the same variant is an eQTL for gene X. That means that there's a linear association between gene expression of gene X and the allele, like let's say risk allele count at that variant. So this might suggest that, okay, maybe this gene is somehow involved in disease and maybe that association is specific to brain tissue. But we can also have the situation where the exact same variant will be an eQTL for gene X in a different cell type. This is a great example of co-regulation. So this means that gene X across two different cell types are co-regulated because they share an eQTL. But let's say an oracle tells us that there is actually no functional basis for this variant in this cell type causing disease. Let's say the oracle has in fact told us that the first row is correct, where the eQTL in brain is sort of the way that genetic variation is mediated to cause disease. All right, now let's just take one more example where we have muscle tissue. It's not the same variant that's an eQTL for gene X, but it is a variant in LD with the other variants that I've been talking about. And this also counts as co-regulation. So effectively, co-regulation, meaning shared causal variants or variants in LD, all make it complicated for us to infer what might be the causal tissue and can drive up associations, much analogous to SNP-based association. All right, so right, like I just said, this is very analogous to the need for fine mapping of GWAS variants. So for example, you might see after running a GWAS in a locus, there might be a great number of genome-wide significant variants, but we actually don't know which is the causal variant. And of course, probabilistic statistical fine mapping methods have been created, such as FineMap and more recently SUSY, and even functional versions of these methods have been produced, which ultimately seek to provide us some posterior probability or even credible set, of variants that might truly be involved rather than just statistically associated. So the method that we've created and that I'm going to talk about today, the goal of it is to identify causal tissues in the presence of many tagging tissues in a data set such as GTEx. So on the left, I'm taking one of the subpanel figures from the LDSC SEG paper, so Finucane et al. 2018 Nature Genetics. This is where they used specifically expressed genes to identify or to at least make annotations for variants that are nearby to those genes and nominate disease-critical cell types. So you see for BMI, there's this big peak for all these central nervous system tissues. And of course, we know that the brain has a strong component in regulating BMI. But as far as which specific cell type or cellular population even in the brain that is responsible for this association is not as well understood. And so ideally, we would want some sort of method that can parse this signal and give us a better understanding and interpretation of what might be the causal tissue or specific cell type. And again, it's very difficult because these are all strongly correlated in all of their expression and co-regulation values. All right, so to help us start to address this problem, we wanted to acquire association statistics that are at the level of genes and tissues. So again, we're not talking about the variant level, so we're not talking about necessarily GWAS variant fine mapping, but we're talking about if we had an association statistic for a gene-tissue pair and how that pair is associated with disease. So TWAS, or transcriptome-wide association studies, is a perfect example of a method that gives us such an association statistic. And this is something that we'd want to parse across highly co-regulated tissues. So here's just the schematic of how TWAS works, so we're all on the same page. So the first step is to get an eQTL cohort, such as GTEx. So for example, you have gene expression measured in the same people that you have genotypes, and then you perform some sort of multivariate linear regression to get the joint fit effects across SNPs to build what are called gene expression prediction models. Now, using these models, you can impute gene expression into a larger cohort of individuals. So let's think about our GWAS cohort of individuals. Let's think of like 500,000 people in UK Biobank. There's no way it's feasible to measure gene expression in that many people by current standards and expenses. And so instead, we can predict what the gene expression might be based on what we know about the genetic component of gene expression. Of course, there's a differentiation between total gene expression and the genetic component of gene expression. So again, because we're trying to follow how genetic variation in the DNA is propagated through gene expression to affect disease ultimately, it's all right that we're talking about only the genetic component, and we're not capturing other sources of variation in the gene expression. So in any case, once you have genetically predicted gene expression across the individuals that you would have in your GWAS cohort, you can perform, let's say if it's a case control trait, some sort of differential analysis where you say, is there a significantly greater or just say different, maybe less, expression levels in cases versus controls. Of course, you could have a quantitative trait that's continuous and look for a correlation. So I want to provide now an empirical example of why inferences from TWAS can actually be quite complicated if you were to do this across different tissues with an example where we quite well know the ground truth. So while TWAS is, like I have here, an approach to find associations between gene and a disease and can be conceptually thought of as the cisgenetic correlation between gene expression and disease. So in effect, it's a polygenic colocalization. It's so where a colocalization would be what I showed in the cartoon a few slides ago. Does the variant that we're finding in GWAS overlap with the variant that we're finding in the EQTL study? This is just like a multivariate version of that, where if we're asking, do multiple variants that we're finding that affect gene expression also coincide with signal that we're seeing in the GWAS? In any case, if you were to do a TWAS on cholesterol at the SORT1 gene locus, which we know regulates cholesterol via liver tissue, you would actually find that you can get larger TWAS association statistics, meaning chi-squared statistics, or let's say absolute value of a z-score, in cell types that are not the liver. And so this is just like one example of where the inference is sort of confounded or complicated in a way because there is co-regulation, and this result would wrongly suggest, for example, that salivary gland is the causal tissue. All right, so what we identified in our analysis of GTEx data is that TWAS association statistics are actually proportional to the amount of tagged causal effects due to co-regulation, or rather, this is what we should expect. And so what we observe is that the association statistics, or chi-squares, are proportional to the estimated amount of co-regulation between tissues. So this is very analogous to how GWAS chi-square statistics are proportional to LD scores, how much correlation there exists between genotypes, which has been shown in LD score regression and is a way that we can make causal estimates of heritability. So here we're taking that same sort of idea. We can derive a similar model that's based on what we know about SNPs and heritability to incorporate instead what we know about genes and tissues and the genetic component of gene expression and how these additively contribute to a complex trait or polygenic disease. And again, we see this trend because TWAS statistics not only include the direct causal effects of genes and tissues on disease, but tagging effects of co-regulated genes and tissues. All right, so now I'm going to present our method and our simulation results. So in order to estimate co-regulation, which is an very important component of this method, if you're not accurately estimating co-regulation, you're not actually understanding how the TWAS statistics that you're getting are related to one another. This is like how accurate your LD scores would be if you're trying to estimate heritability from a GWAS. So the first two steps of estimating co-regulation are the same as the first two steps for running TWAS. So we can estimate co-regulation using gene expression prediction models and a reference panel. So again, step one, let's say we're using GTEx data, we're learning gene expression prediction models, which are from multivariate models, linear models of joint fit effects of SNPs on gene expression. Then we can impute gene expression into not necessarily a GWAS cohort because we've shown that it's sort of robust over very different sample sizes, but we could do something smaller that's more efficient, like 1,000 genomes. From that, we can sort of make a reference map for what is the correlation between genes and tissues, or gene tissue pairs, I'll say, across tissues. So then the next step is to be able to estimate a co-regulation score for every gene tissue pair. So again, for every gene tissue pair, we're getting a TWAS chi-squared statistic. So it's important that we know across tissues how this statistic or this gene tissue pair is co-regulated with each other tissue, including itself. And that's because, so in the schematic here, I'm showing a couple of genes in green on the right, because again, co-regulation can also happen because of EQTLs and LD. Therefore, not only do we have to think about how the same gene is co-regulated just across tissues, but we need to think about how the genes near it also may share EQTLs or have EQTLs in LD. And so in that sense, for every gene tissue pair, we take the sum of the squared correlations of genetically predicted expression, so this vector W that I've shown in step 2, across individuals. For the genes within, let's say, a cis locus, empirically what we use is ±1 megabase, and then we compute this with each separate tissue in turn. So as a result, what we create is this big matrix where on the rows we have all of the gene tissue pairs that correspond to the output from TWAS, and then on the columns is just the number of tissues. And there's sort of like a block diagonal structure to that, where the diagonals are not exactly 1, again, because we're also involving what is the squared correlation with nearby genes. All right, so the generative model that we thought might fit this data, which we've then tested in simulations, is the following. And we call our method tissue co-regulation score regression, or TCSC, and it estimates tissue-specific contributions to disease. So the generative model explains how the expectation of the TWAS chi-square statistic, which again is across gene tissue pairs. So you could imagine if this is a fully like non-sparse vector, you'd have maybe 20,000 genes and maybe 50 GTEx tissues. So that would be the number of points that you have in this regression. It should be proportional to the GWAS sample size, as GWAS summary statistics are taken as input to TWAS. Then the next part describes what I was showing on the previous slide of how we model co-regulation scores. And I'll put a slide is coming that shows more of like a matrix format of this and a little bit less of a mathematical equation format. So in effect, the TWAS chi-square is proportional in some way to these tissue co-regulation scores. And the way that it's proportional is via this slope. And the slope here is defined as the tissue-specific heritability of gene expression. What that means is actually what is the variance of the phenotype that can be explained by genetically predicted expression in a particular tissue. And this is a multivariate regression, so we are estimating one value of h squared GET for every tissue that we're considering in our data set. So ultimately, our method determines that a tissue T prime causally contributes to disease if genes with high co-regulation to that tissue have higher TWAS chi-square statistics than genes with low co-regulation to the tissue. All right, and then we can also expand this model to partition the genetic covariance between two traits. So instead of regressing TWAS chi-square statistics on co-regulation scores, we can regress products of z-scores between two different traits. And the same model holds, except then we're estimating a contribution to covariance. So here's the matrix format of this model. First, we establish this vector of TWAS chi-square statistics across all of our gene tissue pairs. Then we bring in our co-regulation score matrix, which is independent of the GWAS, like independent of the trait that we're looking at. It only depends on the tissues that are present in our model and the genes. So effectively, you would use the same matrix again and again and again, unless you're looking at a different set of EQTL data. So if you were not looking at GTEx, you'd have to recreate this. And so every cell in this matrix represents the co-regulation score between the gene tissue pair that's described in the lighter blue or green on the left with every tissue in turn. All right, and then as a result, this is the vector that we are estimating, which is the heritability of the disease or the variation of the disease that can be explained by genetic expression in each tissue. So the dimensions of this vector match the number of columns on the matrix. All right, so then we applied, we tested out how our method works in simulations. We used the TWAS simulator framework from Nick Mancuso's lab at USC. So ultimately what we are describing a genetic architecture where we dial in an amount of co-regulation across tissues and across genes and a certain heritability for the trait that we're looking at. And ultimately, we can look at three important metrics to see how our method is doing: power, type 1 error, and bias. So for power, I want to specifically point to the EQTL sample size of 300, which is the most representative of GTEx data. And so you can see we have about like 72% power for that EQTL sample size, which is great. We've done this over 1,000 simulations, so that is the 95% confidence interval that you see. Then in the middle for type 1 error, we are asking what proportion of the time out of these 1,000 simulations do we incorrectly identify that the tagging tissue in the simulation is the causal tissue. And specifically in our simulations that I show here, there's 1 causal tissue and 9 non-causal tissues in a single model. All right, and then bias on the right can be broken down into the bias over 2 quantities: the estimated tissue-specific heritability for the causal tissue versus the estimated heritability for the non-causal tissue, which should be 0. So you can see that as EQTL sample size increases, we're doing better on this estimate where the truth is 5%. And of course, noise in the EQTL model, which is synonymous with low power, low sample size, means that we are underestimating this heritability because we're just not capturing all of the genetic variation that is actually involved in the trait. And then in the gray bars, which are very hard to see because they're very tiny, but they're to the right of each of the purple bars, is the heritability estimate for the non-causal tissues, which hovers around 0. We also show that our method has substantially higher power than the RTC colloq method, which I introduced in one of the earlier slides from Ongen 2017 Nature Genetics. And this, again, one of the reasons that we think that this is the case is that this method, the RTC colloq method, takes as input genome-wide significant variance. So it's already very dependent on things like the heritability of the trait and the GWAS sample size. But it also relies on the fact that you need tissue-specific EQTLs in order to identify your causal tissue. And if that's not the case, that method is not able to parse the signal and instead just applies a penalty that is sort of uniform across the tissues that are co-regulated with one another. So it's not really able to take one tissue out of a group and say this is the most likely causal tissue. All right, so now as far as results on real traits, we applied TCSC to real gene expression and trait data. We built gene expression prediction models across 48 GTEx tissues, only keeping heritable protein coding genes. So essentially we use GCTA to estimate the heritability of each gene based on cis SNPs, and then retain the ones with like p-value less than 0.01 and positive heritability, and also ones that were protein coding. So then we analyzed 78 independent European GWAS summary statistics with a large average sample size. And as a result, at 10% FDR, TCSC is able to find 27 causal tissue disease pairs. I don't have this on the slide, but it's able to identify 30 causal tissue trait pair, like covariance results. And ultimately, it increases the specificity of known tissue disease associations, which I'll get into some examples in the following slides. All right, so our main figure result is the following. We have this heat map of traits on the y-axis, GTEx tissues on the x-axis. The dagger next to brain cerebellum indicates that this is a meta-analyzed tissue because one of the parts that's important to follow in this method is that each tissue needs to have the same EQTL sample size. Otherwise, it really throws off the false positive rate of the method. And so therefore, we had to meta-analyze several different brain tissues in the cerebellum provided by GTEx in order to get to that sample size that we were able to analyze in the other tissues, which was 320. The value that I'm plotting in this heat map is what we call pi t prime. This is equal to the tissue-specific heritability term that I've been showing, which is like the h squared GET. But we divide it by the common SNP heritability because we want this value to be comparable across the traits that we've looked at. And the common SNP heritability is the ceiling on what we can estimate with the TCSC model because there are no, we're only looking at common variants in the gene expression prediction model. So there can't be more variation in from the genetic expression than there is genome-wide from these SNPs. All right, so effectively I've starred the ones that have FDR less than 5%. The ones that don't have stars are between 5 and 10% FDR. And I can point out a couple things. One is that whole blood is identified as the causal tissue for the blood cell quantitative traits, which isn't surprising. Liver is identified as the causal tissue for several lipid traits, which is not surprising. I've highlighted 3 different tissue trait pairs, which are sort of interesting. And then I'll go into some discussion of them on the following slides. So first, we find that aorta artery is the most likely causal tissue within our data set. So there's an asterisk on this statement. So because I'm talking about causality, we have to keep in mind that the tissues we're identifying are more likely to be proxy causal, right? They are the best tissues in the model that TCSC can look at to say that there's the most evidence for this tissue. So an absent tissue that is actually the correct tissue that TCSC doesn't know about could in fact be the correct tissue. But since it doesn't know that, it cannot make that inference and effectively say the correct answer that none of these tissues are causal in this data. So this is an interesting result. Aorta artery is a causal tissue for glaucoma because high blood pressure is a known risk factor for glaucoma. We found that the esophagus muscularis is a putative causal cell type for FEV1 over FVC. So this is a lung quantitative trait. It means how much can you expel from your lungs normalized by the total capacity for air in your lungs. And then another example is that we found heart left ventricle is a causal tissue for platelet count. So let's talk about some of these results. So the association of esophagus muscularis with FEV1 over FVC might seem a little strange because we have lung tissue in the GTEx dataset. However, my method was not able to pull up lung tissue as a more likely causal tissue than esophagus muscularis. So what is going on here? So I dug a little bit into where the samples for the GTEx tissues were coming from to sort of make sense of what might be happening. And so in GTEx, they sampled lung tissue from what's called the inferior segment of the left upper lobe, which is highlighted in yellow. However, and this is part of the connection that I'm trying to make with this trait, is that smooth muscle cells are actually in a different part of the lung. And these smooth muscle cells are well known as far as regulating the strength of one's ability to exhale in terms of contracting the airways. And this is also associated with asthma and other, I would say, like lung traits or diseases. Now, if we look at where GTEx sampled the esophagus muscularis from, they sampled it from just above the gastroesophageal junction, which is where the esophagus connects to the stomach. So the interesting part is that this region actually has a lot of smooth muscle cells. And so an important thing to note about our method is that since we're doing this, we've applied this to GTEx tissues. Now, if you consider that every tissue has a different cell type composition, if the wrong tissue has a greater percentage of causal cell type within it, our method is probably likely to say that that tissue is the most likely causal tissue, rather than another tissue that has a lower percentage of the correct cell type. So what I'm thinking here is that potentially the lung tissue that GTEx sampled just didn't have enough of the correct cell type within it. And so my method is saying, well, there's this other tissue that looks better, but probably just because the cell types that are represented in that tissue are closer to the true causal cell type. So this is a big caveat of the method, and that's why we're kind of excited to apply this to better data sets than GTEx. Of course, the limitation is that you need a large EQTL sample size, and doing this in cell type level data or single cell level data really puts you at a disadvantage in terms of the number of donors you can get. But on the other hand, maybe we can compensate for that for the number of cells, as there have been a lot of methods development recently, especially from my PhD lab, in terms of identifying EQTLs in a very robust way from single cell data without pseudobulking. All right, so the other example I want to talk about is the aorta artery in glaucoma. In GTEx, there were no eye tissues or cell types sampled. So if we look at what segments of the aorta GTEx actually took their samples from, surprisingly, they took it from this entire red segment from top to bottom. So there's actually presumably a lot of heterogeneity in these samples, which makes it even more challenging to understand sort of what's going on here. But then if we take a look on the right about how the aorta is connected to the vascular system that leads to the ocular cell types, it's possible that there's just a lot of shared cell type properties. And so even though the tissue might not make a lot of sense, again, I'm saying that cell type composition could play a large role in what we're seeing. And this really tells us that we need better, more precise cell level data to make the best sort of inferences. Let's go on to the next one. Okay, oh, it froze for a second. Let me go back. So I also made the claim that TCSC was able to increase the specificity of known tissue disease pairs. So what I mean is that in GTEx, for example, there are 3 different types of adipose tissues. There's breast adipose tissue, adipose subcutaneous, and adipose visceral. Specifically for HDL, TCSC was able to say that adipose subcutaneous is the most likely causal adipose tissue and did not find any causal signal from the other 2 types of adipose tissue. And interestingly enough, it seems like maybe it has something to do with a mechanism involving adiponectin based on a literature search. But again, these are only computational results and would require further validation to put more confidence behind them. So, the next thing that we think we've increased the specificity of is for waist-hip ratio adjusted for BMI, where again, we have these 3 adipose tissues. Signal from subcutaneous seems to be the most likely causal tissue for this trait. And I want to say it's not just a coincidence that the subcutaneous adipose came up for both of these traits. We looked at the effectively the distribution of our results across the different adipose tissues, and it's not like adipose subcutaneous just has a lot more power than the other 2 tissues. They have very similar distributions of heritability estimates and p-values, meaning the standard error that we're computing in our model is not telling us that we have a lot more power for one versus the other. Finally, for brain tissue, so we know that the central nervous system is highly involved in BMI. Our method picked out that cerebellum is the most likely causal tissue, which seems a bit strange because the cerebellum is associated with motor function. However, in a brain-specific analysis that we did, where instead of a joint model across all of the GTEx tissues, we only did a joint model across the brain tissues, just in hopes of getting more signal out of the residual co-regulation. Because every time you add more tissues, it's harder to find residual signal that points to a specific tissue. And so we knew that we have this prior knowledge that central nervous system is important for BMI, so we felt justified in making that choice to eliminate non-brain tissues. In that analysis, this is where I had the asterisk for amygdala, we found that the amygdala was also associated or causally associated with the brain, which is interesting because the amygdala plays a role in appetite regulation. So this could very well make biological sense. But I want to talk first about really the cerebellum and what might be going on here. So again, I'm going to make the argument that cell type composition plays a large role in the associations that we're finding. So in previous work, it's been shown that specifically expressed genes in glutamatergic neurons are enriched for BMI heritability. We know that glutamatergic neurons are more prevalent in the brain cortex. So why are we not finding any causal link to the brain cortex with our method? It's an open question. We don't really know. But it's interesting that if you do look at the path that these glutamatergic neurons take, you can see that some of the circuits, so specifically this blue one that I've highlighted here, actually points to the cerebellum. So if you think that like maybe there's regulation that is mediated via the cerebellum, even though it originates in the cortex, maybe there's something about the sort of like the order in which this regulation happens, which leads us to pick up on the cerebellum, but not the cortex. That being said, it's also really hard to know based on the GTEx samples for cortex versus cerebellum, what are actually the cell type compositions that we're seeing, and maybe there's some confounding happening at that level. All right, so we compared our analysis on real traits to a couple other methods, just to do some traditional benchmarking and to make the point that our method is a lot closer to fine mapping the causal tissue than just identifying associated. So what we did is every pair of tissues separated by a black line indicates two highly co-regulated tissues in GTEx. So on the left is of each of these pairs is the tissue that we found is the causal tissue from TCSC, and on the right is the tissue that's most highly correlated with it in terms of EQTL effect sizes. If we looked at RTC colloq and then the LDSC seg method, which is performing stratified LD score regression on specifically expressed gene sets across tissues, we see that these methods are not able to distinguish between these pairs of tissues like our method is. For RTC colloq, you just see this wide global pattern of association where there's really like no semblance of cell type specificity happening. For LDSC seg, you do see some sort of diagonal, but still within these pairs of tissues, they receive very similar enrichments. And that's because the definition of specifically expressed genes for this method is a differential expression analysis of genes in one cell type versus the rest in GTEx. And so by definition, the annotations that you're getting for two very closely related cell types are going to share most of their SNPs. So it's not surprising that these estimates are very similar. Now, one might think, okay, maybe there's something artificial about the way that TCSC is saying one's causal and one is not. So what might be going on here? To answer this question, we went back to simulations and we asked what happens in TCSC when the model assumptions are violated. So which assumptions am I talking about? A big assumption of our method is that the gene disease effect sizes are IID, like identical and independently distributed across genes and tissues. Even though there's co-regulation, which means that genetic variation affects genes and tissues in a way that's highly correlated. And this is consistent with the SNP-based analog, right? We assume that SNP disease effects are IID across the genome, even though there's a lot of LD between SNPs. So in simulations, what we did is we duplicated the causal tissue. And what we hope would happen is that instead of TCSC saying one tissue explains 5% of the heritability and the other explains 0, we would hope that it would be able to say 2.5% for one, 2.5% for the other. And this is exactly what happened. So this is good, but again, this is a violation of the model assumption. But still, when this does happen, the model performs as we would hope it would. And this means that this is an attractive property of TCSC because when we do plan to apply it to more high resolution data sets such as cell type level and single cell data, you're getting a lot more co-regulation between your genes and tissues just inherently by the nature of the cell types being more similar to one another within a tissue, for example. And when you start to have these model violations that, oh, like one cellular population isn't actually that different from a second one, especially in terms of its effect on disease, you would hope that TCSC splits the heritability instead of introducing some artificial sparsity. All right, and then the last real trait result that I want to show is applying cross-trait TCSC. So this is using TCSC to partition the heritability, or sorry, the genome-wide covariance of two traits or diseases. And so what we've shown here is that the two traits of interest are BMI and red blood cell count. And effectively what I'm showing in the plots is in the red bars, these are the tissue-specific heritability estimates from the single trait analysis that I've been talking about this whole time. The green bar is the same thing for the second trait. And then the blue bar is the covariance that is explained by the tissue. So ultimately what this shows is that our method found that genes in the pancreas on the left, the ones that tend to have a positive effect on, or like that increase the risk for BMI, also decrease the risk or red blood cell count that an individual might have. On the other hand, if we looked in brain tissue, specifically the substantia nigra, there is a positive correlation between, in the sense that genes that increase BMI risk are also increasing your likelihood of having larger red blood cell counts. All right, so as a conclusion, we've shown that co-regulation scores and TWAS statistics can be used to infer causal tissues, underlying disease heritability, and covariance. TCSC is able to identify new biologically plausible tissue disease pairs. And finally, TCSC might be more informative if it were applied to better data sets, such as dynamic EQTL data sets, where instead of steady state, which we're looking at in GTEx, that's probably not the best avenue to study disease effects, because that's not representative of the data set. For example, with autoimmune diseases, you need stimulated immune cell types to be able to see the effects, not just resting cells. It would be great to look at case control EQTL data again, because we're looking at healthy samples in GTEx, same reason I just gave. And lastly, single cell gene expression data sets. The more resolution we can get, the better inferences we think we can make. Although we'll probably take a hit on power, but we think that the method will still perform reasonably well. So with that, I'd like to acknowledge my mentor in my postdoc, Alkis Price, and other members of the group. This method we have as a preprint on bioRxiv. And as I said earlier, since I just started my lab at UCSD, I'm recruiting grad students and postdocs. So feel free to check out my lab website or contact me if you want to discuss anything further. I'm happy to take questions now.
Hi, thank you very much for that fantastic talk. So I was wondering, like, when you compute this score, like you kind of use all the genes with the EQTL. And I was wondering if you could try to take the subset of the genes, like informed by like a causal gene mapping or like a rare variant exome sequencing analysis to kind of like increase the specificity of the gene disease relevance?
Yeah, this is a great question. And this is something that one of my colleagues in the lab, Ben Strover, suggested, like kind of partitioning the genes based on some sort of function, like you would have in like a stratified LD score regression framework. I just haven't had time to implement that, but I think it would be super interesting to do it, because I think we could hone in on a regulatory process where the signal to noise ratio is a lot better, and that actually helps us identify the causal tissue a lot more powerfully.
Yeah, I think like interpretation of the co-regulation of those kind of analysis may have more interesting insights if you narrow down the specific genes.
Yeah, definitely.
But
so
beautiful
that's
work.
An immediate next step. Yeah, thank you.
Thank you.
Hi, Carl DeBoer from UBC. Great talk. First, I want to say it's really cool that you found that aorta glaucoma connection, because the aorta is like the pathway through which blood pressure is regulated.
Right.
Through the baroreceptor reflex. So that's really cool. So my question is about whether you can deconvolute the bulk data into single cell data, and then you can use like the fact that some cells are in more than one tissue to learn more cell type specific information.
Yeah, definitely. That's a fantastic idea. Some of my colleagues in Alkis's group have been working on deconvolution of GTEx data using mouse single cell data, and then they've done some sort of like TWAS related analysis and like gene level fine mapping. So there's effectively like a data set that I could use to do that very thing, and then try to figure out how it changes our interpretation of which cell types we're identifying as causal. So yeah, thanks. That's a very reasonable path forward.
Hi, my background is in neuroscience, and so I was really surprised to see that connection with the cerebellum.
Right.
And when I saw that slide, I was just on my phone looking up. I saw that cerebellum has a connection to the hypothalamus, which is extremely important for feeding and satiety and body weight. And there's a couple papers on PubMed saying nobody else has looked at this, but we think this might be important. Have you thought? Did you know about that?
I did not know about that, but it's really interesting that you say that. I'm going to write it down.
I'll connect with you after.
Yeah. And I'll just add that I think so we have the hypothalamus as a GTEx tissue that we've looked at. However, we've only looked at it meta-analyzed with other like limbic systems. So it's been meta-analyzed with the amygdala in the main analysis and one other limbic system tissue, which I'm blanking on at the moment. But effectively, like if the signal is strictly from one of those, now that we've meta-analyzed it, we've kind of destroyed that and diluted it. So that's probably why we're not seeing it. And then for other reasons, I think that tissue has a lower sample size than the other brain tissues, and I'm going to blame it on that fact of why we didn't pick it up in the brain-specific analysis. But thank you so much for that comment.
Hi, very nice talk. My question, it's more like a clarification maybe. So the way you're doing this is, so you have a GWAS SNP, and then you have the EQTLs that tell you the expression, and then you're using co-localization to figure out what tissue, and then further on you are trying to, account for the fact that things are correlated. Do you need the TWAS thing? Can't you just use the GTEx? And how much does the TWAS actually help in terms of identifying? Because you cannot go beyond what you're actually getting from GTEx and Juvadis. Do you need the why is the TWAS actually useful?
Yeah, so the TWAS is where we get the disease level information from, or the complex trait that we're interested in. Because we don't have phenotyping data for the people in GTEx, nor nearly at the sample size that we would need to identify disease level effects. So the TWAS is how we pull in the GWAS summary statistics.
But you already have the GWAS loci, right?
Yeah, but TWAS doesn't just look at genome-wide significant loci. So it just looks at all of the spectrum of association statistics. So it uses GWAS chi-squared statistics and the EQTL and sort of like does a polygenic co-localization of that. It's just like a wrapper for the way that the GWAS is integrated into the EQTL data.
I see. And then going to single cell data, I mean, the only thing you'll get is the gene expression, the cell types stuff. But how will you get the genetic component of it? Or will the single cell has to have the genetic, it has to come from a population level data, right?
Otherwise, yeah, so we would have to be able to have genotypes matched for the donors that are in this single cell data set.
I see.
The tricky bit is that then you have to invoke a random effect model for your donors because you have many cells that are coming from the same person that are not independent observations. But in that sense, the EQTL model can't simply be a linear model. And that's never been done in TWAS before, really. So that would be a methodological innovation in that sense. But yeah, the unfortunate thing is you do need the genotypes, which makes this method a little bit less accessible than something that just looks at total expression. But methods that look at total expression can't get the genetic component precisely modeled. You can only guess as to the nearby SNPs by those genes as being relevant.
Cool, thanks.
Hi, interesting talk. Can you comment on phenotypes that are known to be associated with the multiple cell types or tissues?
Yeah.
I'm actually quite surprised to see that the DBP or the diastolic blood pressure in your second last slide are implicated in only one tissue. I believe it was aorta artery. But from my understanding, there are many GWAS loci associated with the blood pressure are implicated in many different tissues, such as heart, kidney, and even the adipose tissue. So I just want to see your comment or the why your method only implicates just the one single tissue rather than pointing out multiple likely causal tissues.
Sure. That's a good question. And what was the trait again?
The DBP,
DBP.
yeah,
Okay,
the
right. Yeah.
diastolic blood pressure.
Right. So we're only finding aorta artery, as you can see. As far as like kidney, we don't have that in GPEX. We do have the two different heart tissues, the heart, like atrial appendage and the left ventricle. I don't think it made it onto here because we were also looked at systolic blood pressure, but because they're highly genetically correlated and we wanted to look at only independent summary statistics, we removed it. But I do recall that systolic blood pressure also had heart as a causal tissue that was identified. So I don't know if it has to do with like the power of the summary statistics or the fact that maybe like the data from the aorta just looked a lot cleaner and less heterogeneous than the heart tissue. But there's nothing that's enforcing that only one tissue or a few tissues is identified in TCSC. So I want to chalk it up to power, but I think there's probably like a nominal significance for that tissue. It just didn't make the FDR.
Okay, thank you.
Yeah.
I guess I had a related question. I mean, so you're not enforcing it only to return one, but it seems like there's some bias towards it. And then you were trying to show some simulation that it seemed to be performing reasonably in that sort of specific example. But do you have more either theoretical results or empirical results in like these more high resolution settings that this model should be able to handle situations where we have such that we would expect potentially a lot of finely defined cell types to all be relevant?
Yeah, so we have simulations that I haven't shown here for the sake of time, but that are in our preprint where we have multiple causal tissues per trait. It's not necessarily like higher, it's the same like tissue framework. So it's like pretending that multiple GTEx tissues are causal. It's not that we've done it at like the cell type level or the single cell level at all yet, but hopefully that answers your question just a little bit. So the intuition behind those simulations is that we wanted to see like how big of a power loss do we suffer when we have like 3 or 2 causal tissues that are highly co-regulated. And there's like a power loss, but there's still like like I would say, I think it's something like 50% power to identify all 3 of them as causal. So based on that, I wouldn't be too concerned that that we're seeing like singular tissues pop up as causal. I think it just has to do with the power and the heterogeneity of the sample. And just larger sample sizes will help us identify more of them.
Okay, thank you.
Yeah.
Hi, thanks for the talk. I was wondering, like, if you have any sense of like what should be the size of the cohort with like when these methods become applicable for single cell studies. Like you showed this example of the 1K data set, I think. So it's already 1,000 individuals.
Was it this one?
This one exactly. So do you think like 1,000 individuals are already enough to like apply our method for single cell, or do we need like a lot more?
Can you get 1,000 individuals for a single cell? I mean, that sounds, so the paper that I'm referencing here, like Nathan 2022 Nature, that has 250 donors and 500,000 memory T cells. So I'm thinking that ballpark should be more than fine.
Okay.
And for bulk tissue, I mean, yeah, you want to have like at least several hundred, and in simulations, which again are like the cleanest way to look at this, like we're finally getting unbiased estimates at 1,000 individuals, which means in noisy real data, you'd probably want more than 1,000 to get unbiased results, but that's really hard to do by current standards.
Yeah, I think like this one, if I'm not mistaken, I think they have like, it's called 1K because they have 1,000 individuals and 1,000 cells for each individual.
I have heard of that data set, yeah.
So, but yeah, like it's different for different individuals, and you still have like only 1,000 cells per individual. So the representation of different cell types could be very different. And things like that.
Do you know if there's genotyping data available for 1K?
Yes.
There is. Okay.
Yes.
Great.
Yeah.
Cool. Thanks.
Okay. Thanks.